The question we will be looking at is in mice, resveratrol increases training-induced mitochondrial biogenesis. However, this effect is not present in humans. Why? To answer this question, we looked at mitochondrial protein as our mass, with mRNA as our inflow and protein degradation and breakdown as our outflow. Mechanisms regarding protein degradation and breakdown have not been discussed in class, and therefore we are assuming that outflow is constant. The increase in mitochondrial protein in mice is therefore due to an increase in inflow, and more specifically, an increase in transcription of mRNA. Inflow is driven by PGC1 alpha mediated transcription.
We formulated our hypothesis based upon the following points. Resveratrol activates SIRT1 in mice, but not in humans. SIRT1 activates PGC1 alpha mediated transcription and increases in mitochondrial biogenesis, and PGC1 alpha activated in mice therefore increases the training-induced mitochondrial biogenesis in mice. In essence, we hypothesized that resveratrol activates SIRT1, and SIRT1 activation is higher in mice as compared to humans. Therefore, PGC1 alpha activity and training-induced mitochondrial biogenesis is increased in mice and not humans.
As discussed in class, the transcription of PGC1 alpha is influenced by several factors. PGC1 alpha phosphorylation is increased via the activation of KNPE and subsequent activation of P38, as well as by the activation of AMPK. Furthermore, PGC1 alpha is deacetylated via SIRT1, which is activated indirectly by AMPK and increases in blood epinephrine concentration, ultimately activating transcription and mitochondrial biogenesis. Despite being influenced by several factors, the research we have examined only found increases in PGC1 alpha transcription and resultant increases in mitochondrial protein via resveratrol through the SIRT1 pathway. We will therefore focus on SIRT1 as a mechanism by which PGC1 alpha transcription is activated within muscle cells.
Based on our hypothesis, we looked at two different types of research, the effects of resveratrol in mice as well as in humans. A study done by Marie Legouge looked at how resveratrol improves mitochondrial function and protects against metabolic disease by activating SIRT1 and PGC1 alpha. The metabolic effect of resveratrol was evaluated by a cohort of male mice. They were given a dose of 200 or 400 milligrams per kilogram per day of resveratrol, administered in either a chow diet or a high-fat diet for 15 weeks. The test in mice demonstrated that the SIRT1 activator, resveratrol, induces PGC1 alpha activity by facilitating SIRT1-mediated deacetylation, resulting in an increase in mitochondrial function, translating into increases in energy expenditure and aerobic capacity, consequently protecting the mice from obesity.
A review completed by Williams and Gerd in 2012 investigated the control of skeletal muscle mitochondria biogenesis and attempted to identify pathways leading to the activation of SIRT1 and PGC1 alpha. The study investigated the influence of exercise, calorie restriction, and pharmaceuticals on SIRT1 activation. The study was completed in order to apply the findings to obese and overweight populations and to direct future health studies and directions. First, the findings showed that SIRT1 activity is accompanied by increases in PGC1 alpha transcriptional activity. The researchers also found that there is uncertainty in humans if resveratrol directly influences SIRT1. However, resveratrol may be indirectly activating upstream targets of SIRT1. Resveratrol has been shown to activate AMPK, which may induce increased SIRT1 activity in mice. However, this mechanism is unknown. There is no clear mechanism that shows that resveratrol is directly exerting an effect on SIRT1 in humans. Overall, there is no direct link between resveratrol and SIRT1 activity, and SIRT1 is not conclusively affected by resveratrol in human studies. In conclusion, the data demonstrated by Legouge support our hypothesis that in mice, RSV activates SIRT1, leading to increased PGC1 alpha. This consequently increases exercise abilities and aerobic capacity due to the PGC1 alpha mediated increase in transcription of mitochondrial protein. The paper by William and Gerd does not identify a mechanism that conclusively states that resveratrol activates SIRT1. Therefore, resveratrol would not exert an effect on PGC1 alpha and does not induce mitochondrial biogenesis in humans. Gerd identifies that the bioavailability of resveratrol in humans may be affecting the activation of SIRT1 and could explain the lack of activation of SIRT1 by resveratrol.